Hello, Assalamu alaikum. In this video, we will understand about the DNA damage. So, we will also understand about the repair of the DNA damage. But what is the DNA damage? Let's understand about that. So, basically, every time DNA damage, but the mismatch, repair work regularly on checkpoint during the cell division when cyclin and CDK activity stop and stimulation on the basis of requirement. If the it will not work correctly, so the DNA damage will not repair and progression of the cell division will progress lead to cancerous cell production. What is the DNA? So the DNA is the long molecule, the macromolecule. It is known as the deoxyribonucleic acid. And in this way, the here you can see this, the DNA is the basic structural and functional unit of the cell. So the DNA is important for the production of protein. So in this way, here is the DNA will basically transcribe into the mRNA, and this mRNA will lead to translation of protein. So this adenine always bind with the thymine with the hydrogen bonding in the nitrogen base, while the backbone is the phosphodiester bond with the ribose sugar will occur, and the DNA is the double-stranded. And when the mRNA will produce during the transcription, lead to translation and protein synthesis. That protein is basic structural and functional part of the cell, and if the mismatch of the nitrogen space and the gene alteration will lead to protein synthesis will not mature, and in this way the disease will occur. Remember that things. Anyhow, we will understand about the common cause of DNA damage now. So this is a chromosome contain a histone protein 60% and 40% DNA in the form of chromosome, and the DNA expressed due to the acetylation or methylation to suppress. Oh, okay. Here is common cause of DNA damage due to several agents like the oxygen radicals, ionization radiation, and chemotherapeutics. The single strand break is due to the free radical of oxygen, O2 oxygen, and hydroxyl free radical or hydrogen peroxide. In this way, the single strand break, while the ionizing radiation and chemotherapeutics collectively will lead to interstrand cross-linked. In this way, the DNA damage, that is the interstrand cross-linked will occur, will lead to gene basically mutation. The gene mutation will not work properly to synthesis of that proteins will be not correct, and that is why the disease will progress. UV light and polycyclic aromatic hydrocarbons will lead to bulky adducts interstrand cross-link will occur. In this way, you can see the tetrate of the nitrogen base will cross-link. The base mismatch is due to the replication stress. The replication stress is due to the during the cell division. The DNA replication will stress, and in this way the mismatch repair will not occur. The base mismatch will occur. Let's understand about the process of the DNA damage. Here is the ribose sugar with the nitrogenous base, the guanine for example. So the reduction oxidation reaction will lead to damage the DNA. How? This is the carbon number 6 contain basically carbon. In this way the hydroxyl ion, the radical will bind to produce water. So in this way the DNA contain carbon, the free radical. This free radical basically bind with the nitrogenous base with the carbon to basically to bind the carbon to carbon linkage will occur for the again oxidation. This is the reduction to lead to oxidation, and here is the nitrogen which that will lead to free radical. So the electron and proton will again bind, lead to damage the structure of the DNA. In this way, here you can see this is the oxidation will lead to basically electron and proton will bind in the nitrogen side, will lead to normal and free radical clear will lead to 8,5-cyclo-2-deoxyguanosine formation will occur. So this is the reduction oxidation will lead to DNA damage. So here is the promoter and suppressor and CDK, cyclin and C-mice or RAS gene. The promoter will promote the CDK and cyclin or the oncogene activation. When the suppressor damage the cyclin and CDK, our activation to uncontrolled cell division, cause proliferation and DNA mismatch, repair not work. Because the promoter will promote the oncogene part, promoter will inhibit the suppressor when the promote, but the suppressor will suppress. But the DNA damage will lead to suppressor will not work to inhibit the promoter, will lead to uncontrolled protein synthesis, CDK, cyclin, and C-mice and RAS will lead to uncontrolled cell division due to DNA damage and progression of cancer. Let's understand about here is you can see this is basically the promoter and the suppressor gene working. We will understand about the normal cell division and then we will understand about how cancer will occur due to the DNA damage. This is the cyclin and CDK2 will lead to progress the S phase, synthesis phase, will lead to cyclin A and CDK2 will lead to progress the G2 phase and G2 phase work with the cyclin A and CDK1. This G2 phase will lead to progress the cell division, the mitosis through cyclin B and CDK1 will lead to prophase, metaphase, anaphase and telophase and cytokinesis will occur. And in this way, the cell will replicate into two cells and this daughter two cells sometime the G0 phase will occur like neurons and other cells, but some will like stem cell which that will lead to a progress for further cell division for proliferation. And in this way the checkpoint is important, the checkpoint protein if not synthesis. So in this way the uncontrolled progression of that like cyclin our expression due to the DNA damage. If the DNA damage not work not create, so in this way the normal physiology of the cell will work. The single and double stranded break will occur due to the ionizing radiation through direct effect lead to damage and free radical progression, while the indirect effect is the water will ionize in the form of proton free radical and electron and hydroxyl free radical, and on the other hand hydrogen peroxide H2 H3O plus. And in this way the basically it will lead to also cause the DNA damage. Antioxidant for the DNA repair, we will understand how the DNA will repair. So the ionization potential of the antioxidant is more and bond dissociation enthalpy will occur. In this way here is the free radical like hydroxyl free radical or reactive oxygen species which that will combine with the antioxidant. In this way the donation of the electron for the pairing and accepting of the electron for normal free radical to the normal molecule or atom. In this way the electron donate the single transfer of electron and will lead to radical intermediate with oxidant plus proton. And in this way you can see this is the free radical which that will accept the electron or donate the electron for the pairing will lead to neutralize not reactive. So on the other hand the proton, for example radical and antioxidant complex plus or antioxidant will lead to combine together, and in this way the activity of the reactive oxygen species will not work, the hydrogen atom transfer. The free radical and antioxidant here is you can see the donate of electron and unpaired electron to pair formation will lead to reduction and free radical will remove. The oxidation of molecules damage and DNA, RNA, protein and plasma membrane, lipids, even all cell body. Antioxidants prevent oxidative stress, because this is all oxidative stress due to the reactive oxygen species. So remember that things. Basically, we have discussed about the DNA damage and how to repair. Thanks for watching. See you in the next video. For this and bye.